everyone, and welcome to another episode of GenLive. On today's show, we'll be discussing the microbiome. I'm Juliana Lemure, science writer at Gen, and I'll be your host today. I am joined by two of my colleagues and GenLive regulars, John Sterling and Kevin Davies. Hi, John. Hi, Kevin.
Hello, Juliana. Hi, everyone.
And we're joined by three very special guests today. Emily Drabant Conley is the CEO of Federation Bio. Although a newcomer to the microbiome field, she's no stranger to leadership roles at large companies in cutting edge and highly competitive spaces. Before joining Federation Bio, she spent a decade in a leadership role at 23andMe, a company that endured controversy while making a name for itself in direct-to-consumer genomics. Today, we'll talk to Emily about starting a new microbiome company and what Federation Bio is working on. Hi, Emily.
Hi there.
Unlike Emily, who's recently come to the microbiome field, Sarkis Mazmanian has been thinking about bacteria for a long time. Having done his graduate work in bacterial pathogenesis, he's focused his lab's research at Caltech on the gut-brain connection. With seminal papers showing the link between the microbiome and Parkinson's and autism spectrum disorder, Sarkis will tell us about how that subset of the field is going, and also what's going on at the two companies he founded, Axial Biotherapeutics and Symbiotics. Hi, Sarkis.
Juliana, hi, everyone.
And Sonia Timberlake is the Senior Vice President of Research at Finch Therapeutics. Having done a PhD in the lab of microbiome veteran Eric Alm at MIT, Sonia too has been in this field for some time. Before her current role, she spent some time at Ab Vitro and Juno, and today she'll tell us about what's going on at Finch Therapeutics, one of the older companies in this space. Thank you for joining us today, Sonia. So the microbiome field may be at an inflection point. With over a decade of research and data built up, the concept of using bacteria to treat many common diseases now appears poised to deliver the first clinical results. With research and drug development thriving in areas from neurodegenerative diseases to skin disorders to antibacterial resistant infections, the promise of the microbiome is growing rapidly in both academic labs and industry. That said, some are still skeptical, as is common in a scientific field that's still cutting its teeth. Today, we'll discuss the new frontier of microbiome research in academia and industry, recent progress, clinical potential, and future challenges. And we would like to hear your questions too. So please put any questions that you have for our guests in the Q&A section of the Zoom, and not the chat. So in the Q&A. So we're going to start with the basics, and Kevin, I'll pass it over to you.
Thank you very much, Juliana. Welcome, everybody. Sarkis, a warm welcome to you. What is the microbiome briefly, and when did it first begin to be thought of as a platform for drug development?
The microbiome refers to the collective genomes of the bacteria, viruses, fungi, and single-celled eukaryotes in our gut and skin and many environmentally exposed surfaces. Microbiota refers to the cells themselves, and so the functional components of the microbiome. So we often call it the microbiome because that's how these bacteria are identified and characterized is through genome sequencing or even sequencing of specific genes like the 16S ribosomal RNA gene that identifies the organism or the organisms. But of course, we'd like to think about function in terms of what our gut bacteria, viruses, fungi are doing to promote health and disease. And so, that's the collection of organisms, about 100 trillion bacteria in our gut, which dominate our microbiota, most of the bacteria that live in or on us or in our lower gastrointestinal tract. In terms of the field itself, like many current fields, the roots of the microbiome or microbiome research or gut bacterial research really extend back over 100 years. In the first identification and the knowledge that these organisms have been part of our bodies. But in terms of I'd say the modern age, of research in this space. I would say about 20, 25 years ago was some of the most pioneering work, really obviously just scratching the surface, very unsophisticated compared to today's research, but really trying to understand how these organisms are networking with our bodies. A lot of this obviously was in mouse models or other preclinical platforms, but thinking about how bacteria can tune or educate, if you will, the immune system as it develops, again, in research settings. And it's really a change, a departure in the way we viewed our association with bacteria or microorganisms in general. I think many of us, grew up thinking that microbes were these insidious little creatures. They want to just make us sick. And we studied bacteria and other microorganisms from a pathogenesis or infectious disease standpoint. But I think that's maybe one of the take-home messages that we've learned over the past couple of decades, is that we're actually in many ways dependent on the function of specific organisms for our health and well-being.
Thank you very much. We'll come back to you in a few minutes. Sonia, let me bring you in. What's different about microbiome-based drugs compared to the typical process of drug development?
Yeah, and it's one of the things that I'm most excited about in this field, because so I think there are a few fundamental differences. One is I think that the products of this field, when we're talking about delivering healthy commensals as a drug, I think there's a lot of evidence that the safety and tolerability profile of a drug that's comprised of healthy commensal bacteria is much better. And I think that has a lot of important implications, not only for, the kind of vulnerable populations that we might be able to go into, but think about intervening earlier in the disease course, think about, treating children, or maybe even someday, preventative medicine, right? So my 2-year-old's had 3 rounds of antibiotics already. Wouldn't it be great if after each of those rounds of antibiotics, wouldn't it be great if she hadn't had oral antibiotics, but wouldn't it be great if we could sort of retrain her microbiome organ as her immune system and all of her systems are developing. So that's one, I think a huge difference. And I think, we're beginning to see that borne out as some of these drugs are going into the clinic. I think another big difference, and this is a bit more hand wavy at this point, but the true believers in the field feel this way, is I think we have the opportunity to treat the root cause of diseases. And so, you contrast a bacterial therapeutic, for example, for ulcerative colitis with a systemic anti-TNF. That's a systemic immunosuppressant to treat sort of a localized, GI inflammation. And if the disease pathogenesis is driven by something that's happening in the microbiome, then using a microbiome targeting therapy, or better yet, healthy commensal bacteria that might be missing from that ecosystem, I think by treating the root cause of the disease rather than having a systemic, immunosuppressant, I think that, not only might it be safer, but, less side effects. And I think just it's more satisfying to treat the root cause of the disease rather than suppressing the symptoms. So those are the, two of the main differences that I see in terms of how we approach drug development. This is sort of like biologics was 25 years ago, right? There's manufacturing challenges. There's so much unknown biology. There's so much, unknown, at the interface between this new organ and your gut epithelium, and how it interfaces with your whole, all the systems of your body, right? There's just so much unknown. And so I think one of the fun challenges that we deal with as we're moving through clinical development is, how do we start making the FDA, generating the FDA guidance for how such drugs should be regulated, right? How do we generate all of the new procedures for manufacturing, for talking about these drugs? And so I think there's a lot of analogies to biologics 25 years ago when a lot of these things were unknown.
Very interesting. Thanks so much. John, over to you.
Emily, welcome. Thank you for coming, and welcome back from a well-earned vacation after a 1 or 2 year hiatus like the rest of us. So I have 2 questions for you. One, why did you jump from genomics to the microbiome? And two, how did your experience at 23andMe, and in particular, working alongside the company's CEO, Anne Wojcicki, prepare you for being the CEO of a microbiome company?
Good questions. Kind of similar to the analogy that Sonia was making about biologics and how that's sort of relevant, kind of the nascent biologic field 25 years ago has some parallels to where we're at with the microbiome. I spent, previous to taking the helm at FedBio, quite a bit of time in the genomics field, and had the privilege of watching that space grow kind of in the early days. So when I joined 23andMe in 2010, genomics tools had certainly progressed and had kind of already gone through a hangover of overpromising at the early 2000s that genomics were going to kind of help us find better drugs and solve diseases, and then a hangover when that didn't materialize in that particular time frame. And I got to sort of see that the upswing of that happen over the last 10 years or so, where not only did the tools get better from a sequencing standpoint, but the data sets got bigger, which turns out to be really essential in leveraging genomics for a target discovery and target validation standpoint. And what I foresaw was that the microbiome was sort of where genomics was 10 years ago, that the tools have improved. The microbiome is orders of magnitude more complex than the human genome, and has the potential to just massively impact our health in ways that we don't completely understand yet. And that was really appealing to me to be at sort of what I view as kind of the beginning of how the microbiome is now being leveraged from a therapeutic standpoint. And I love one of the things that I enjoyed in my time at 23andMe was the sort of disruptive nature of the platform, and I think that similarly, microbiome, like nobody's cleared a microbiome therapeutic through the FDA yet, so there's a similar kind of disruptive nature here that was really appealing. And in terms of the experiences, I had the great fortune of working alongside Anne Wojcicki for a long time. She's a terrific leader. That 23andMe was not without its challenges, including regulatory challenges with the FDA, innovating in their business model, growing. The company was about 30 people when I joined and about 600 when I left. And so I felt that those experiences were great training grounds for taking the role that I'm in now.
Yeah, that's for sure. Hey, would you describe Federation's technology platform, including microbial immunotherapy? And how does it work?
Yeah. So there's actually two platforms that are totally distinct, although they can be leveraged together. And the first is building large, diverse, complete consortia of commensal bacteria, so naturally occurring bacterial strains. And the real differentiator there is that we're building consortia that are extremely large, so over 150 different strains of bacteria as a single drug product, and they're focused on a lead indication in a kidney disease. The second platform is microbial immunotherapy, as you've mentioned, and there we're taking advantage of really some of the unique properties between the human immune system and the microbiome. As Sarkis was pointing out, that the microbiome trains our immune system in really, really fundamental ways. And so what we're taking advantage of there is skin microbes that are commensals that exist on the skin, and engineering antigens of interest into those microbes, which then drive antigen-specific T cell responses. And it turns out that the flavor of bacteria that you use, the species of bacteria, drives what kind of T cell response you get downstream. So whether that's CD8 T cells or T regulatory cells. And so we're leveraging it in both directions, in the case of oncology with CD8 cells, and in autoimmunity with T regulatory cells, where we're putting in antigens of interest, and then seeing pretty incredible effects in vivo in animals, where those T cells that are naturally derived can go and perform the very powerful biology that T cells are capable of.
So a little more talk about how Federation designs, discovers, and develops communities of potentially therapeutic microorganisms.
Yeah, and I think this is an interesting topic. It'll be fun to hear Sonia and Sarkis' thoughts on this as well, that the microbiome is so vast, and if you look at any two people, the species of bacteria between them in their microbiomes might be quite different. But if you look genomically at the function, the core functions that those bacteria perform, there are similarities. There are core functions that the microbiome produces. And so we've really, rather than looking kind of species by species, we have taken a very genomics focused approach of thinking about function and what are all the kind of key biological niches that we want to fill in a healthy human microbiome. And so in the lead program that we're focused on, it's a kidney disease that is caused by excess absorption of oxalate, which is something that we eat. Oxalate is found on spinach and chocolate and a lot of foods that we consume every day, and most of us excrete it. But we've designed a consortia there that degrades oxalate. And a lot of the design principles are around understanding the synergistic relationship that the bugs that are degrading oxalate are producing formate. And so then we need some formate reducers because that can feed back and inhibit the oxalate degradation. So we've got this kind of system of, we've designed like these core bugs that are reducing oxalate, producing hydrogen and reducing formate. And then we have a whole bunch of bacteria around those as sort of a supportive backbone that would perform the kind of core functions of a microbiome. So that when we transplant that as an organ, the way Sonia was talking, this really is a living organ, that it can function completely and engraft.
So talk a bit more about that idea of using many bacteria in one drug. But again, a little bit more about what's the thinking behind that?
Yeah, I mean, I think this is, to me, this is a big part of what's exciting about this space. And I sort of feel like in the early days of the microbiome, there was more of a focus on kind of one particular species of bacteria as having special properties and thinking about like, oh, okay, this particular strain is going to really have an effect. And I was talking to a KOL who said, yeah, but you know, if you've got a patient who has a super dysbiosed gut, like somebody with IBD, and you put in one or two healthy microbes, it's like putting a fish in a tree, right? Like those healthy microbes, they don't have the ecosystem, the support that they need to actually perform their function. And so for us on the consortia side of our platform, really thinking about like what we've learned from FMT, from fecal matter transplant, that when you transplant a whole ecology of microbes, you can look at those patients 5 years post-transplant, and a majority of the bacteria you transplanted are still there because they were part of an ecosystem. And so that's what we've stayed focused on, is like, okay, we need to understand mechanism and we need to choose bacteria that perform the mechanism that's going to give us the therapeutic effect, but then we have to couple them with everybody else that can help all of those bacteria engraft and function.
Yeah, that's like a real holistic approach. Well, Juliana?
Thanks, John. So Sarkis, you started your training in bacterial pathogenesis. What prompted you to move over to the microbiome?
It was just that it was an open space. And so again, this was about 20 years ago, before the microbiome was cool. And there just weren't that many people in researching, all these bacteria that we knew, again, for many decades lived in our intestines. And I just saw this as a unique opportunity. I loved microbiology for many years prior to that, and I wanted to stay close to the field. But I also want to do something different, sort of, venture off into the unknown. And it was really just the fact that it was just this wide open space, that made it both challenging and exciting. And of course, I couldn't have ever predicted that the field would grow so rapidly and so vastly. Of course, it's quite gratifying that it has. And just to see all the new technologies and new perspectives that have come into the field, again, prior to which, I think we were just taking a very simplistic approach. And now, obviously, in terms of both computational approaches, coupling multiple different omics technologies, and as well as the research that's being done in humans, I think is really catapulted the field forward just in the past few years. But again, for me personally, and just a small note, that it was just the fact that it was just unknown.
Yeah, I always tell my students when I'm teaching, like when I started my PhD, the microbiome was not a thing. Like it just was not a thing that we learned about, or to really even, it was brand new at the time. So cool. So you've started 2 companies, at least, I think, in this space. How's it been to wear both the academic and industry hats at the same time?
Really fun, all right, and a great learning experience as well for me, being a basic scientist, working at a research institute with no hospital, but thinking about medically oriented problems and human health, but again, from from a preclinical and basic science perspective. And so our lab is really focused on novel discovery, trying to understand interactions between microbes in the immune system, the metabolic system, more recently the nervous system. The companies have allowed me to then translate that work into both development as well as into clinical trials that have been already accomplished and are ongoing, to really test these hypotheses, right? So I think part of the skepticism I think in the field is because we have relied so heavily on these preclinical models, on mouse models, like the biologics field, like the gene editing field and other fields as well. But I think the microbiome offers us a unique opportunity to move into human research more rapidly than traditionally speaking. So if you were thinking about small molecules and novel chemical entities, I think the safety and toxicology issues that arise with novel drugs are largely circumvented by using organisms that come from humans. And I think as Sonia was referring to, I think the FDA is quite bullish about the space and really, feels that in many ways the safety and tolerability studies have been done over millions of years because the bugs come from the human gut. And so again, the companies have allowed us to take our ideas and our concepts and ask, is there clinical relevance to them?
Mm-hmm. Yeah, got it. So you've been focusing quite a bit on the gut-brain axis. Can you explain, how is it that bacteria in the gut can affect what's going on in the brain?
Yeah, again, something I learned or am still learning is just the vast highways of communication between the gut and the brain, the number of different conduits by which these organs communicate. And of course, we're just focusing on these two. Obviously, the brain the body is an intertwined system. And I can pontificate on how we train scientists to be very focused on particular disciplines and organ systems or diseases, but we need to really think about the body, and all the different tissues and all the different organs, all the different cells, both human and microbial and others that are talking to each other. But just in terms of the gut and the brain, clearly there's neuronal connections, right? So the vagus nerve, which connects many organs, including the gastrointestinal tract to the brain, the circulating immune cells, about 70% of our immune cells at any one given point in time are in our gastrointestinal tract. So again, being sort of educated and trained, as we've always talked about, by the microbiome, and these cells can then circulate. And some of them can get into the brain or near enough to the brain to affect brain physiology, and in some cases like autoimmunity, actually induce pathology or protect from pathology. There's obviously circulating cytokines and small molecules, neuropeptides, and neurotransmitters. Many of our neurotransmitters are heavily focused in the gut. So about 90% of the serotonin in our body is in our gastrointestinal tract. 50% of the dopamine in our body is in our gastrointestinal tract, much of which is either produced by bacteria or its production is regulated by gut bacteria. So we're still trying to understand those relationships of how these really important brain signaling molecules, why they're made in the gut, and how does the production of or the regulation of these molecules in the gut affect brain physiology. And the final area is one that we've become quite interested in, is the direct transit of molecules, small molecules from the gut into the brain that then control various aspects of cellular physiology, brain function, namely connectivity and brain activity, as well as their effects on complex behaviors. And so work from many groups, including ours, has shown that there are potentially hundreds of microbial molecules, molecules produced only by bacteria, not molecules that our bodies produce that are regulated by bacteria, but of strictly microbial origin that wind up in the brain. And so the bugs don't have to be in the brain, but their signals are sent to the brain through these small molecules. And again, we're barely even scratching the surface and trying to understand what those molecules are and certainly what they're doing. I think this is really ripe for really thinking about traditional drug discovery, drug development platforms and concepts, but applying them to natural products instead of synthetic compounds being taken orally that may wind up in the brain, but thinking about how we can leverage these small molecules that are produced by bacteria that over evolution have essentially found their way into the brain and affect brain function. So I think that's an area that we are quite excited about.
Okay, building off of what you just said, how can all of those mechanisms, how can that be druggable? Is it make more metabolites, soak them up? I mean, how does that work for a drug?
Yes, all of the above, right? But obviously, first, basically trying to understand what the molecules are, what they're doing, and whether or not high levels are detrimental, or the absence of particular metabolites or small molecules have negative effects. So obviously, this is based on discovery, based on again on leveraging of preclinical models, but clearly in a number of classical neurological conditions, neuropsychiatric, neurodevelopmental, neurodegenerative conditions, there's an imbalance of small molecules in the microbial origin in the brain compared to healthy individuals. And so this again gives us an opportunity to either supplement missing small molecules, either by giving the micrometabolites directly, or by giving the bacteria that produce those metabolites. But the converse is also true, is that there's an elevation of particular micrometabolites that correlate with negative outcomes, such as anxiety or autism. That's been chronicled by us and by other groups as well. And so, and this is one of Axial's platforms, is to think about these potentially neuroactive, maybe even neurotoxic molecules, and how one may drug them. And the approach that Axial's taken is essentially to sequester these small molecules in the gut and have essentially these absorbents, these gut-restricted, non-absorbable molecules that are given as drugs that specifically bind classes of microbial metabolites produced in the gut that may have these negative effects. And by doing so, the molecule is excreted along with the microbial cargo that it binds to, effectively lowering its concentrations in the serum. We know that in humans, potentially in the brain. We know that just from mouse studies, not in humans. But by lowering these harmful molecules, essentially by drugging the gut, one may perceive to get beneficial effects. And of course, this, in many ways, I think is overcomes one of the biggest challenges in dealing with neurological conditions. Is the current medications have really a high bar, because the burden is to get these molecules into the brain in the right doses in the right regions without side effects. And of course, we know that many anxiolytic drugs, antidepressants, other drugs for neurological conditions have immense side effects, because it's really only a fraction of the molecules that we take orally that actually get into the brains. But of course, as you just asked and we discussed, microbes have figured out this pathway, figured out these highways and how to get their molecules into the brain. All we have to do is, I guess, is figure out what they are, what they're doing. But by drugging the gut and not having to get drugs into the brain, I think it's really a much more tractable approach.
Okay, terrific. Thank you so much. I'm going to pass it over to Kevin, but before I do, Kevin, I just want to say we have some great questions coming in from the audience. So please keep the questions coming and just throw them into the Q&A box. Okay, Kevin, over to you.
Yeah, thank you. Sonia, tell us a little bit about your company, Finch, how it got started, your role at the company, and the company's overarching goals.
Sure, yeah. So Finch was started by Mark Smith and some other co-founders, and Eric Alm was involved. And they had first started Open-Biome, which was the nonprofit stool bank back in like 2012-ish. Mark started out of his grad school lab. And that nonprofit grew to provide treatments for over 50,000 C. difficile patients. They worked out a whole regulatory paradigm with the FDA to provide these safe treatments. And then as they were doing that, they saw that fecal microbiota transplant, so the act of sort of replacing a diseased microbiome organ with a full, complete healthy microbiome organ, is a great tool for research. And all these researchers all over the world, there are over 300 just on clinicaltrials.gov right now, are using FMT as a tool to ask, is the gut microbiome capable of affecting a clinical phenotype in diseases as diverse as IBS, Parkinson's, immune oncology response, like just a whole world of possibilities. And so they looked at that and said, hey, let's start a company that can really translate those insights from, academic studies with FMT into real drugs for patients. And so what Finch is really based on, so Finch develops live biotherapeutic products, so human gut commensals to treat disease. And the basic premise is to really rely on fecal microbiota transplant data as a way to say, like, yes, the gut microbiome can have a positive clinical effect in this indication. And to do reverse translation, look at, which bacteria are colonizing the patients that respond, which functions, which metabolites, as a way to sort of unpack the mechanisms behind that clinical response. And so Finch, I think, is unique in that we have two product strategies, two technology platforms for drugging the microbiome. One is what we call a complete consortia, which is similar to what Emily described, in that you don't want to rely on just like one bacteria, right? Yep. If you have a disease where there's a very severe dysbiosis, or where you don't fully understand all of the complex mechanisms that are at work, the best thing for patients might be to provide them with a sort of a healthy, full community. And so that's, hundreds of species where we've optimized the manufacturing process, a GMP manufacturing process, to deliver those in an oral lyophilized pill. And then a second technology platform, which is more the targeted approach, where we have single strains of bacteria that have been isolated from donors that were used in the clinic as part of that bigger consortia, single strains that are targeted for having specific functions, specific impacts on the host, ability to produce specific metabolites. And we grow those up in pure culture in big, big batch fermenters, and deliver those as oral products. So I guess we look at this like, we have sort of a suite of tools, and depending on the indication, what does the patient's microbiome look like? What do we know about the mechanisms of disease that are involved? What do we know about the size of the patient population and its specific needs? We can deploy, these two platforms sort of tailored to the indication of interest. Maybe one more thing about Finch. We're developing in a lot of different areas, which I can talk about in a moment. But we are the most far along with our C. diff program. So we're the first company to complete a positive, pivotal, like a registrational study. And that was our phase 2, and we're starting a second pivotal this year. And so that was like with this oral lyophilized complete consortia for the treatment of C. difficile. But we just see that as like the very first step in a really, really exciting field, where there's possibility in everything from autism to immune oncology.
Well, for folks who are a little bit newer to the field, why is C. diff such a big problem?
Yeah, so C. difficile affects, I think, about 500,000 Americans a year. And it's not just like you get the runs. People have, crippling diarrhea, like they can't leave the house. Actually, Mark was inspired to start Open Biome because one of his family members, a young med student, contracted C. difficile, and for 18 months was on and off like 6 successive rounds of antibiotics, had to like drop out of school. So it's really a crippling disease. And I think about 30,000 people a year actually die from C. difficile. A lot of people that contracted are older, immunocompromised people who are hospitalized for another reason, receiving antibiotics. Antibiotics is a major risk factor for C. difficile. And there are a lot of conditions that also predispose you. For example, people with IBD, and they don't respond as well to some of the treatment options out there. So it is a big problem, and unfortunately, as we continue, globally to use more and more antibiotics, more and more people worldwide become susceptible to this disease.
Are there other drug targets in your pipeline besides C. difficile?
Yes, we have a number of candidates, and I should just correct myself. I said registrational trial. We have to complete 2 trials before we're approved in C. difficile, and we've completed one of those 2. So we have a couple of phase 1s starting in the next 15 months. One is in chronic hepatitis B virus. One is in autism spectrum disorder. And then additionally, we have some programs, some product candidates in development with Takeda. One is a consortium, a targeted consortium for ulcerative colitis. And we just expanded that partnership to include Crohn's. And then we have a bunch of exciting, pre-clinical discovery work. And I'm the head of research at Finch. So that's more.
And we come full circle.
Yeah.
Very good. Thanks so much. John, passing it over to you.
Yeah, I just want to say we're a little bit more than halfway into the program. This has been really interesting and engaging. And you're all very enthusiastic about what you're doing. So thank you. 2 ways you can tell is, number 1, we got a lot of questions. Number 2, every time you answer something, you raise 5 other questions. And that's great for a journalist. So thank you. But Emily, what are the biggest challenges in getting a microbiome drug into development at this current point?
Getting it into development or getting it approved?
Getting it further along in development and then getting it approved.
Yeah, I mean, I think that there's certainly the microbiome field faces a lot of the same challenges that other fields face. I think there are some unique challenges that we face in the microbiome, given the complexity of the system. So like, for instance, if you're developing a small molecule, you don't have to worry so much about the diet of what patients eat every day, because that's not going to affect the way that the small molecule is metabolized in their body. But for those of us working in the field of the microbiome, what you eat feeds the bacteria in your microbiome. And so if you've got a patient who is a vegan versus someone who is, a carnivore, they are likely to have natively very different microbiomes, and they may respond very differently to your therapy. So there's more variables that we are contending with in the microbiome. So one is kind of environment, diet, heterogeneity. Another is the complex interactions between the microbes. And I think this is probably what's exciting to all of us that are working in this space, that there's a lot to understand, and these interactions, there's synergy between the bugs, what a certain set of microbes, what they eat and then excrete becomes a food source to another set of microbes, all living in the same ecosystem. So there's a lot of variables here. And I think for us, the focus has been on really trying to laser in on mechanism, on places where we can say, all right, there's not just an association between these bacteria and this particular downstream phenotype, but we actually understand the mechanism of this is what is causing the disease, and this is what the function that these bacteria perform that could then alleviate it. And so trying to make that connection really tight, that's been a big focus. And then I think translation, as Sarkis was talking about, we have some cool tools in the field of the microbiome. So one of the things that we can do is we can actually study animals that don't have a microbiome. They're called germ-free animals. So these are mice that are born with no microbes in or on their bodies. And that gives us the control to put in the microbes that we want. It also isn't that translatable to humans, because we're all colonized with all kinds of microbes, and there's the real life system is a lot more complicated. So mapping that translation is something that I think we all need to be pretty focused on.
And once again, can you talk a little bit more about your lead product candidate and what else do you have in the clinical pipeline?
Yeah, so the lead program in enteric hyperoxaluria, which is essentially a disease characterized by chronic kidney stones. So these are patients that are getting kidney stones over and over and over again. These are calcium oxalate kidney stones, and that eventually can result in end-stage renal disease. There's about 250,000 patients in the United States that are living with this condition. And a lot of these patients have like an underlying malabsorptive disorder. Many of them had Roux-en-Y gastric bypass surgery. So their their intestinal tract has been kind of radically altered by this surgery. And as a result, when they eat these oxalate containing foods, they're absorbing the oxalate into the blood and then forming these kidney stones. And there's currently no approved treatments. The main way that physicians handle this is by telling these patients to drink more water, eat less oxalate containing foods, eat more calcium. So it's which not surprisingly is not really effective. So there's a big opportunity and a big unmet need. And what we're excited about is that we have evolved microbes that perform this important function of oxalate degradation. And we've been able to isolate those microbes. And so the platform at FedBio is essentially isolating microbes from feces, from healthy donors, and then selecting those microbes and growing them up, kind of as Sonia was describing, growing them up in the lab in pure culture so that we can really closely control what exactly is in that consortia.
Wow, fascinating. Before I turn over, it reminds me, you're saying that we're these collections of all these microorganisms. The first time I heard a lecture on this topic, the scientist who ended, I forget his name, he said, oh, so be sure when you go to bed at night to say goodnight to all of yourselves. So
Yeah.
I think we're onto something there. Juliana?
Okay, thanks, John. So Sarkis, I'm going to ask you a question about engrafting versus kind of passing through the gut. So, some microbiome drugs are designed to engraft and become permanent members of the bacterial community. Other companies are designing them to pass through. And actually, Isabel, this is an audience question, says, as a drug treatment, would you aim for a transient effect or one that would establish permanency? And I guess, what are the advantages and disadvantages to either of those?
Sure. So I think at the bottom line is that we have to figure these things out empirically in the clinic, right? There are obviously advantages to engraftment, as has already been discussed, is that these organisms, evolve in these polymicrobial communities. These microbes don't live in isolation. We study them in the lab in isolation, but that's not how they evolve. And so when you give a consortium of organisms to an individual, you're giving a higher chance of that organism to engraft, assuming that there are still ecological niches that are open for those organisms. And oftentimes, some of the trials to date have used antibiotic or even bowel cleansing to clear the indigenous microbiome so that we can get a better engraftment. So when the organisms are there and they're living and they're thriving, they're producing their molecules, they're finding the right little nooks and crannies in the gut, and they're ramping up the production of these potentially beneficial processes, whatever they are. In most cases, we don't know what the molecular output is, but we just know this community has a beneficial effect. And so, but the shortcoming of that approach is, again, a lot of this needs to be figured out, is what are the long-term effects of engrafting wholesale microbiome into people, foreign microbiomes into people. If there are adverse events, what do you do about that, right? And how do you address that? Of course, again, these are issues that are being tackled actively as we speak. The transient approach, I think, there you're relying on an organism as it's passing through the gastrointestinal tract to produce enough of a particular molecule, or to change the community, or to restructure the metabolic capacity of that indigenous microbiota to then have some sort of beneficial effect. And so, of course, you're asking more from an organism that doesn't engraft. But you're also, I think the trade-off is that you're probably going to have a therapy that's easier to address if there's an adverse event, because you can just, like a traditional drug, you can just withdraw the treatment itself. There's also a dosing effect, right? Because once an organism engrafts, their relative levels are and the equilibrium that those organisms reach are hard to control. So as Emily mentioned, you may be able to use diet or other ways to modulate particular levels of certain organisms. But again, a lot of work that needs to go into that before we know exactly what to give, and then what diets people should be on. But again, it's very hard to control the levels of organisms once they've been grafted. Again, they're all relying on each other, and again, finding their ecological niches, and they'll get to a certain equilibrium. Whereas if you give an organism that's transient, then you can dose in a more traditional way. That you can give, again, higher doses if you want more efficacy, lower dose if you want to deal with certain side effects. So I think, again, there's trade-offs to both approaches, but I think the answer is still all in the clinic.
Okay. We've had a couple of people ask the same question, so I'm going to ask you, how do you define a healthy microbiome?
It's difficult, right? And so I think for many years, a lot of us, and to this day, we correlate a diversity in the microbiome with health. And there's a lot of evidence for this, right? I mean, there's a lot of studies when people have compared, or studies have compared a healthy population to, a disease population of choice. Let's say many, many different diseases have been studied from a microbiome perspective, because it's pretty straightforward these days to take, stool samples and, using DNA sequencing, get a microbiome profile. And invariably, in most cases, when people have looked, they've seen differences between a disease state and a healthy state. Whether or not those changes, this is sort of said, whether or not those changes are a consequence of the disease state or a driver of the disease state, a lot of this still needs to be figured out. But what many of those studies showed is that diversity in the microbiome correlated with health. And so I think that that's one way of defining health, is that, again, as Emily alluded to, that you have these, multiple different functions, right? So essentially, there's more benefits that the body can reap from having a microbiota that is quite diverse. But as Michael Fischbach tells me, and so he's a co-founder of Federation, that he's seen many, bad pathways, bad organisms in very diverse microbiota. And so, I think we do need to revisit this topic from a more scientific perspective, again, from a more evidence-based perspective. I think that's what has really percolated more often than not, is this diversity. Obviously, one can take a more structured approach, or a more targeted approach, and ask, are there particular pathways that we know correlate with health that are enriched in a healthy state, sort of like butyrate production, for one example, seems to be an area that a lot of people are interested in. And people will correlate certain levels of small molecules, like called short chain fatty acids, which are broken down from dietary fibers by our microbiota, and butyrate is one of those short chain fatty acids, and ask, do people who produce more butyrate, do they tend to be healthier? And there's some evidence that that's the case. There's some evidence that butyrate production and short chain fatty acid production are depleted in the disease state. But again, I think that that's, it's a very coarse measure, right? I think we need to really understand what are the flux of molecules, more than just one pathway. What is really the collection of products that these microbiota are producing. And the other aspect, getting back to the interplay with the host, is how does any one particular microbiota interface with a particular genetic landscape in the individual? It's not one size fits all. I don't think it's going to be one size fits all. I think there's a large element of personalized medicine here, where particular organisms, consortia, pathways would function differently in individuals depending on their genetic landscape. And I think that's something that we still are not really tackling well, I think, in the microbiome community, because it adds such a level of complexity when you think about population diversity at the population scale. But I think it's an important concept that we should all remember, right? Is that, I may have a particular set of organisms that in my body, may be harmful because of my genetic predisposition. In somebody else's body, they may be benign, right? And so I think that's something that we should definitely continue to pursue and study.
Okay, thanks, Sarkis. Emily, a question came in for you specifically that asks, do patients who suffer from the kidney disease that you were talking about have a different microbiome to patients who are healthy or don't have the disease?
They do. And in particular, they appear to have less of some of these oxalate degrading bacterial strains than in kind of the general population. I will say, though, that hasn't been the thrust of our approach. Like, our development approach hasn't been kind of profiling these patients, figuring out what they're missing relative to healthy controls, and then making that a drug. It's been more mechanistically focused. But kind of building on what Sarkis has said, there are definitely differences between patients with disease versus healthy individuals, and some of which are driven by diversity that you can see.
Can I ask a question for Sonia? Several people asked a question about current treatments or treatments in the pipeline for IBS. So we'd love to get your thoughts on that, as well as what is in the pipeline that you know of for autism spectrum disorder, and what would the rationale be for treating that particular disorder?
Yeah, great question. So for IBS, that's not something we have in the pipeline today, but we monitor the FMT literature very closely, and there's been a lot of exciting results there, where there have been, I think, 2 placebo controlled randomized studies, like large studies, treating IBS with healthy donor complete communities, so through this fecal transplant. And they've had, they've met their primary endpoints, and they use the sort of same diagnostic criteria that drugs use. And so we think that those are incredibly exciting. And actually there's a link between these two questions, because what many people might not know is that people who suffer from IBS often have clinical anxiety and depression. And people people with autism spectrum disorder very often suffer really crippling GI pain, constipation and diarrhea, lots of GI issues. And the parents will tell us that those are very correlated with the neurocognitive and behavioral symptoms. And so I think, Sarkis talked a lot about these many links between the gut and the brain. And we think that there's a lot of interesting biology and a lot of interesting new data that supports the effect of gut microbes on your immune system, systemic inflammation, neuroinflammation, as well as direct signaling through the vagus nerve and other metabolites. So to turn to autism, we are starting a phase 1 there in the next before the end of next year, and there we're building on 5 studies that used 5 academic studies that used fecal transplant, people all over the world, including one of our collaborators at Arizona State University, to treat pediatric patients, pediatric kids with autism spectrum disorder, and looked at the effects both on those GI symptoms that I mentioned, as well as their neurocognitive and behavioral scores. And they were really exciting. When we saw this first one study, the first study that came out at Arizona with our collaborator a few years ago, we were like, wow, that's interesting. Can that possibly be real? And then, over the last 5 years now, it's been, successive studies in humans, all of this great mouse work that's uncovered some of the plausible mechanisms that could be driving this. And now, we have all of this, conviction that it's real. And so we're starting our phase 1 next year. First, looking at safety and tolerability in patients as young as 3. And then we're really hopeful to get some of the behavioral and neurocognitive and GI readouts from that and push this out to patients, because, so 1 in 56 kids in the US right now, gets a diagnosis with autism. That affects the whole family. That's a huge number of people. And there really aren't a lot of good treatment options for those families. Like right now, the standard of care is 40 hours a week of intense behavioral therapy. And, despite that, many of these kids aren't able to, have a return to a really normal socialization, and it's a big challenge for those families. So we're really excited about the promise there.
Yeah, you've all painted in your own ways a very optimistic, very exciting picture of the future of the microbiome field. I'm wondering, what are the headwinds? Because no new paradigm gets off the ground. Look at CRISPR, look at gene therapy, look at consumer genetics that, as Emily was saying earlier, these all have their headwinds and challenges and setbacks. And I think there have probably been some clinical trial setbacks in the early days of the microbiome. There's a high profile company where there's allegations of fraud. So what are some of the business headwinds and obstacles that you've got to overcome before to really make good on the exciting research that you've been talking about? Any of you?
I think it's mechanism. I think it's complexity of the system. We're still at the beginning, and there's a lot we don't understand. And there's so many more variables here than with other modalities of small and large molecule drug development, kind of more traditional modalities. So I really think it's understanding and having the kind of scale of data sets that we need and translatable models to understand and make therapies. I'm very optimistic, but I think that's I think mechanism is one of the hardest parts of what we're doing.
Okay, thank you.
And I
Please,
obviously
Sarkis,
agree.
yeah.
I obviously agree with that, right? So getting to basic mechanisms, similar to what you would do with any small molecule drug development campaign, right? Is to understand, what are the receptors, what are the side effects, what are the clinical outcomes. Obviously, as I mentioned, and I think it's just implicit, is just the heterogeneity of the microbiome and how it, it interacts with different, patient population, different genetic backgrounds, right? And so identifying, which are the individuals or who are the individuals that may respond to any one therapy, right? So trying to get that subset that in using some metric, in most cases we don't know of, who would be the responder versus a non-responder, whether it's based on the microbiome profile, whether it's based on their clinical profile, or maybe even their genomic profile. And I think from a business standpoint, obviously there's been a lot of investment in the space, which I think we're all grateful for. But at the same time, the burden of proof is on us, right? It's on the community, right? Not just the academic community, but obviously, the biotech community and industry, is to actually have some wins in the clinic, right? And there have been some high profile failures, and I think they have cast a shadow on this entire space, but I think that's par for the course, right? We're still learning, we're all figuring out, what are the most promising ways to push this technology forward. And as you've already mentioned, and you can see, I mean, there's a lot of reason to be optimistic, but I think there are just some, reasonable challenges that all fields face, and I don't think the microbiome is any different.
Thank you.
I have a question. So one of my classmates became an astronaut in 1987, was 3 times up in the shuttle, was mission specialist. And we'd sit down and talk afterwards. He'd tell me what happened to his body physiologically or otherwise. Have there been any studies on the microbiome in space, that what happens in a micro environment, that when you get back, any lessons to be learned? Or is that still too early in the game here?
I can maybe address it. There have been a couple of studies which show that spaceflight does affect the microbiome, and there's unrelated studies that show that spaceflight affects metabolism, affects neurological functions as well. And now people are still trying to link that, right? So my understanding, I'm not an expert in the field, I just had some exposure being at a place like Caltech, where astronomy is a prominent discipline. Is that there are these early observational studies that the microbiome does change, right, in ways that are not yet predictable but are measurable. What that means physiologically, how widespread that is, how actionable that is, I don't think we know yet. And again, it's not surprising, right? I mean, you're changing the entire environment of an individual, once they're in space, and the microbiome is very sensitive to our changes, the changes that we experience in life. And so the fact that the microbiome is changing isn't surprising. I don't think it should be surprising, but whether or not those changes in any way have a physiological consequence, I don't think that's been answered.
Thank you.
I think we have time for one more question. So, I mean, the word microbiome now, you can find it on yogurts, you can find it on advertising for soap or something, health products. So how do you think the scientific field, the drug development field, should instill confidence in the microbiome drugs when they start coming out and being available, with this word that has become such a buzzword over the past 5-10 years?
I definitely think that's something that this field has to contend with in particular. Sort of a feeling of like, is it woo-woo or is it real science? And with like probiotics as an unregulated space, making a lot of promises, that does not help us. But I think consumers can have confidence that just like all other drug modalities, we will go through the same rigorous development and regulatory process as everybody else, so that when we get to the other side of that we have medicines that are effective. And I think, as kind of Sonia was highlighting in the beginning, I think where there's a lot of excitement and optimism is that because we're in many cases developing medicines leveraging innate microbes that have evolved with us as humans over millions of years, the safety profile is pretty incredible of these therapeutics. And so I think that's something to be really excited about, and makes me really excited for patients. We're developing therapeutics in oncology, for instance, and these are patients that are enduring extraordinarily toxic therapies that have massive side effects. And so if we can make therapies that are effective and that have a very different safety profile, I think that's really exciting.
And I know we're up against the hour, but if I can just add to that comment is, I think that there's a double-edged sword here, because the microbiome, the concept resonates with the non-specialists, with non-scientists. I think the public understands, that there are these bugs that live in our intestines, and our digestive system is important to us, and what we eat is important to us. And so it's not as abstract as other fields may be to the general population. And of course, probiotics are prevalent. You can go buy dozens of different products that are commercially available now. I think the burden on us is to differentiate the existing organisms from the therapeutics, or from the next generation probiotics, if you will, that will come from the human gut, as opposed to dairy products, which were developed, microbes from dairy products, which are the current probiotics, which were developed many decades ago. So I think part of it is the messaging, but part of it is obviously the evidence that we use to support these new therapeutics.
Okay, terrific. Well, that brings us to the end of our discussion and the end of this session of GenLive. I want to again thank Sarkis, Sonia, and Emily for joining us today, and we look forward at Gen to following this field closely in the future. Thanks, Kevin and John and the rest of the Gen production team for making this episode run so smoothly, and thanks to all of you for joining us today. Don't miss our next GenLive in September, where we'll be talking about some of the latest advances in cancer research. I'm Juliana Lemire. Stay safe, and we'll see you again next month.
Thank you, panel. Great job.
Thanks, everybody. Take care.
Thank you. Bye.
Bye.